Imagine this hidden kingdom, like ancient beyond reckoning, doing some of nature's really tough jobs, breaking down wood, even animal hair. And yet, for centuries, we barely even knew it existed. Today, we're pulling back the curtain on these microscopic unsung heroes. The phylum Chytridiomycota, or as we'll probably just call them, chytrids. So, our mission on this deep dive is basically to take a pretty dense chapter from Introductory Mycology and sort of distill the key stuff about these fungi that often get overlooked. Think of it as your shortcut to really getting why these tiny fungi punch so far above their weight. There are some surprising facts and real-world connections that might just make you look at, well, dirt or even cows a little differently.
Exactly. And for a long time, biologists largely ignored chytrids,
Yeah.
mostly because they're just incredibly small and pretty elusive. But their ecological significance is actually profound, and their biology is genuinely fascinating once you start digging in. We're talking roughly maybe 100 genera and around 1,000 known species. And each one is playing a subtle yet absolutely critical role across all sorts of environments.
Okay, so if you had to pinpoint the absolute defining thing, the one characteristic that really sets them apart from all other fungi, what is it? What makes a chytrid, well, a chytrid?
That's a great question, and there's a very clear answer. Chytridiomycota are the only members, the only ones in the entire fungal kingdom that produce motile cells, cells that can move on their own. And that applies both to their zoospores, which are basically tiny swimming spores for asexual reproduction, and their gametes for sexual reproduction. Each of these motile cells typically has just one flagellum, a single whip-like tail at the back that propels it, kind of like a microscopic tadpole, really. Although, just to keep things interesting, a few rare species actually have multiple tails, multiple flagella.
Wow, okay, so swimming fungi, that's definitely unique. What else defines them?
Well, beyond that swimming ability, there are other key things. Their main body structure, we call it a thallus, is often coenocytic.
Coenocytic?
It means it lacks internal cross walls, or septa. So imagine like a long single chamber, but instead of one nucleus, it's packed with many, all sharing the same space. No dividing walls. And these thalli, they vary a lot in shape. Some are simple little spheres or ovals. Others are more elongated, almost like the hyphae you see in other fungi, or even quite complex branching structures. Their cell walls are mostly chitin and glucan, which is pretty standard for fungi. But interestingly, cellulose, usually found in plants, has been found in at least one species. And when they reproduce sexually, the zygote, the fertilized cell, usually becomes a tough resting spore, or a resting sporangium, like a survival pod for harsh conditions. Or sometimes it develops into a whole new diploid thallus.
That's amazing detail for something so small. So how do these biological specifics, play out in the real world, ecologically speaking?
Right, ecologically they are vital. They're primary invaders and decomposers. Think of them as nature's first responders for breaking down tough stuff. Things like chitin from insect shells and other fungi keratin, which makes up hair and nails, and tough plant materials like cellulose and hemicellulose. They tackle the hard jobs. And you find them everywhere. Aquatic habitats, especially freshwater, are hotspots, but also in lots of different soils. Even, surprisingly, in some desert soils. They're much more widespread than people used to think.
Okay, but if they're so common and so important, why the long delay? Why were they overlooked for so long?
It really boils down to their size and the methods we used to use. They're tiny, often needing direct microscopic checks of infected tissues. And the standard ways of isolating most fungi or bacteria, they just don't work for chytrids. Researchers often have to use specific baiting techniques.
Baiting, like fishing for fungi.
Kind of. Yeah, they'll put things like pine pollen, bits of leaves, fruit pieces, even snake skin or cellophane into water or soil samples. The chytrids are attracted to this bait, and then scientists can isolate and study them. It's pretty clever, like microscopic detective work.
That is persistent. So, okay, beyond their big ecological job as decomposers, where else do they pop up? Do they affect humans or our interests, good or bad?
Oh, absolutely. On several fronts. On the downside, some are serious pathogens. They cause real trouble in agriculture. Syncytium endobioticum, for instance, causes black wart disease in potatoes. It can be devastating for potato crops.
Blackwort, right, not good.
Not good at all. Then there's Physoderma matis, which causes brown spot of corn. And Opidium brassicae, it's a parasite of cabbage roots, but maybe more importantly, it's a vector.
A vector, like carrying diseases.
Exactly. It carries several plant viruses that are economically really significant. So it's not just the fungus itself causing harm, it's delivering these viruses too.
Okay, so definitely some bad actors. Is there a flip side? Any good chytrids?
Yes, definitely. On the flip side, some show real promise for biological control. Species in the genus Colomomyces parasitize mosquito larvae. So there's active research into using them to help control mosquito populations. Imagine the public health benefits there.
Wow, fighting mosquitoes with fungi. That's cool.
It is. And in the lab, some free-living ones, saprobes like Blastocladiella emersonii and Alomyces macrogynes, have become really popular model organisms. They're relatively easy to grow and manipulate, so they're great for studying fundamental cell biology, molecular processes,
Hmm.
helping us understand how cells work.
Okay, lab workhorses. Anything else?
Yes, and this is maybe one of the most fascinating areas right now,
Mm-hmm.
the anaerobic chytrids, the ones that live without oxygen. You find these in the guts of herbivores. Think cattle, sheep, horses. They live in the rumen intercecum and play a huge role in breaking down the tough plant fibers in their diet.
So they're helping cows digest grass.
Precisely. They're gut microbes, essentially specialized fungal ones. And researchers are really interested in whether we could manipulate these fungi, maybe make them work faster and more completely. Imagine if livestock could get more nutrition from the same amount of feed. It could improve animal health and potentially lower costs for farmers. It's a really active area of research.
That's incredible. From potato warts to super cows, it's quite a range. Now, you've used some specific terms like thallus and rhizoids. I bet there's a whole vocabulary just for describing these guys, right? Can you unpack some of that chytrid language for us?
There certainly is, and it helps describe their different habits. First, you've got endobiotic versus epibiotic. Endobiotic means living entirely within the host's cells, like Syncytium, the potato wart one. You can't see it from the outside. Epibiotic, on the other hand, means the reproductive bits are produced on the surface of whatever they're growing on, but they have structures, like rhizoids, sunken inside to absorb nutrients. Rhizophidium is a good example of that.
Okay, endo inside, epi on top. Got it. What else?
Then there's how much of the body gets involved in reproduction. Holocarpic means the entire thallus turns into one or more reproductive structures, like Lepidium. The whole thing converts. But in eucarpic forms, only a portion becomes reproductive. The rest, like the anchoring rhizoids, stays vegetative. Chytriomyces does this.
Hollow for whole, you for?
Eu just means true or good here, implying a separation of parts. We also talk about monocentric versus polycentric. Monocentric means just one center of growth and reproduction.
Hmm.
Polycentric means multiple centers, often spread out.
Makes sense. And those rhizoids you mentioned.
Right, rhizoids are these delicate thread-like filaments. They contain protoplasm, the living cell stuff, but no nuclei. Their main job is anchoring the fungus and absorbing nutrients. They can be tiny or really extensively branched, almost like a root system. You might sometimes hear an older term, rhizomycelium, for those extensive systems, but it's used less now because there's just so much variation. Oh, and sometimes, especially more complex ones, you see pseudosepta.
Pseudosepta. False walls.
Exactly. They're like partition-like plugs, but chemically different from the true rigid walls you see in many other fungi. Gives them some flexibility.
That's a great rundown of the terms. So with all these different forms, how does a tiny swimming spore actually grow into one of these? What are the main ways they develop?
Good question. Once a zoospore settles down and forms that protective cyst, there are basically two main developmental routes, depending on what the nucleus does. In endogenous development, the nucleus just stays put inside the original cyst. The cyst simply enlarges and eventually becomes the sporangium, or multiple sporangia. This can be monocentric or polycentric.
Okay, stays inside. And the other way?
The other way is exogenous development. Here, the nucleus actually migrates out of the cyst into a little germ tube that starts growing. Then it divides, and the rest of the thallus forms from that germ tube. This can also be monocentric or polycentric. And if it's exogenous and polycentric, it can lead to either colonial forms where you get clusters of sporangia called sori, or filamentous forms with that extensive rhizomycelium we mentioned, which can just keep growing and spreading.
Right, right. Now, let's circle back to that really unique feature you mentioned right at the start, their ability to move. Those swimming cells. Let's zoom in on the zoospore. What makes these tiny motile cells so special, and what's going on inside them?
The zoospore really is their signature move. Asexual reproduction in chytrids is mostly through these zoospores, produced inside that sac called a sporangium. Inside the developing sporangium, you have this mass of protoplasm filled with nuclei. This whole mass then gets cleaved, divided up into lots of tiny, usually single-nucleated units. Each one develops that posterior flagellum and becomes a zoospore. It's like a little factory churning out swimmers.
And how do they get out? Do they just burst forth?
Sometimes it's quite dramatic. They can get out through one or more papillae. These are like little bumps on the sporangium wall that dissolve to form a pore, and the zoospores ooze or swim out. Or, in some species, there's a specific circular cap called an operculum. It functions like a preformed lid that pops open. Species with this lid are called operculate, and those without are inoperculate. Once they're out, they swim around for a bit, find a good spot, settle down, form that cyst wall, and then germinate to start a new thallus.
Okay, but what about inside the zoospore? You mentioned it's key for classification. What are we looking at?
Right, the ultrastructure, which you see with an electron microscope, is incredibly informative. So they usually have that single posterior flagellum. But interestingly, there's often a second kinetism, or basal body nearby. It's like a dormant, non-functional flagellar base. Inside, you find the usual suspects: mitochondria for energy, microbodies for metabolism, endoplasmic reticulum, lipid bodies for storage. But a really key diagnostic feature in aerobic chytrids is the MLC, the microbody lipid globule complex.
MLC complex. Sounds complicated.
It's a specific organized arrangement. Picture lipid bodies, microbodies, mitochondria, and some membranes all clustered together. It's thought to be involved in using that stored lipid fat for energy while swimming, and also maybe regulating calcium levels, like a little integrated power pack and control system.
Wow. Okay. What else is unique?
Another really unique thing is the gamma particle. It's this small, membrane-bound little blob. We think it probably stores protein. You don't really see it elsewhere. And finally, the ribosomes, the cell's protein factories. They can either be scattered around, or in many chytrids, they aggregate, they cluster together, often forming this really conspicuous cap over the nucleus. We call it a ribosomal aggregation, or nuclear cap.
So these are just simple swimming dots. They're packed with intricate machinery.
Absolutely. They are dynamic, highly organized little machines designed for dispersal and finding new homes.
Incredible. Now, okay, asexual reproduction via zoospores is clear. What about sex? Do these fungi get it on? And with all this variation we've talked about, how on earth do scientists actually classify them? Sounds like a headache.
Well, sexual reproduction has been reported in some, but honestly, for many chytrids, especially the simpler ones, it's either unknown or still a bit questionable. We definitely need more modern studies to really pin it down. But where it has been described, there are a few main ways plasmogamy, the fusion of the cell contents, can happen. One is planogametic copulation. That's basically swimming gametes finding each other and fusing in the water. They might be identical, isogamous, different sizes, anisogamous, or even like a swimming male sperm fertilizing a non-moving female egg. Another way is gametangel copulation, where the entire contents of one reproductive structure, a gametangium, gets transferred into another one. And then there's somatogamy, which is simpler just the fusion of ordinary vegetative parts, like two rhizoids bumping into each other and fusing.
Okay, several options for sex, even if it's not universal. Now, classification, the headache part.
You hit the nail on the head there. It's been described as being in limbo for a long time. Historically, scientists try to classify them based on morphology, what their thallus look like, the shape of their sporangia. But those features turned out to be really plastic. They could change a lot depending on the environment, even in controls lab settings. So relying on looks alone was proving unreliable.
So what replaced morphology?
The first big shift was towards using that zoospore ultrastructure we just talked about. All those internal details, the MLC, the nuclear cap, the kinetism position, turned out to be much more stable and reflected deeper evolutionary relationships. And now, more recently, DNA sequence data has become absolutely crucial. Comparing gene sequences is giving us a much clearer picture of their family tree and how the different groups relate to each other and to other fungi.
The DNA revolution strikes again. So where does that leave us now? What are the main groups?
Currently, 5 orders are generally recognized. You've got the Spizellomycetales, Chytridiales, Blastocladiales, Monobopharideales, and the newest addition, the Neocalamasticales. That's the group with those gut fungi. There's still some debate about their exact placement within the whole fungal kingdom, how they relate to the other major fungal phyla. But the picture is definitely getting clearer thanks to ultrastructure and DNA.
Fascinating how science keeps refining things. Okay, let's make this real. Can we dive into some specific examples from these orders? Really see these concepts in action?
Absolutely.
Yeah.
Let's start with the order Spizellomycetales. Remember, their zoospores typically have dispersed ribosomes, the nucleus is linked to the kinetosome, and sex is mostly unknown. A really cool example here is Rosella allomyces. It's an obligate endoparasite. It has to live inside another fungus, specifically one called Allomyces. And what's amazing is its host mimicry. Its sporangia end up looking almost exactly like the host's sporangia. It chemically senses its host, injects its protoplast, which has no wall, right into the host cell. It develops entirely inside using the host cell wall as its own outer layer. Then boom, it releases its zoospores explosively.
Whoa, a fungal cuckoo. Sneaky.
Very sneaky. Another important one in this order is Opidium brassicate. We mentioned it earlier, the cabbage root parasite. Endobiotic lives inside the cells. Its notoriety comes from being a vector for those damaging plant viruses.
Right, a disease carrier. Okay, next order.
Let's jump to the order Neocalymasticales. These are the anaerobic ones from herbivore guts. They're a big hallmark. Polyflagellate zoospores. Some have over 10 flagella. And because they live without oxygen, their zoospores completely lack mitochondria. Neocalymastics is a key example. They're just crucial for breaking down plant fibers in the rumen, releasing nutrients the animal can use. Those multiple flagella help them actively swim around and find new bits of fiber in that complex gut environment.
Gut powerhouses. Okay, what about chytridiales?
Order Chytridiales. Here, the zoospore nucleus is not connected to the kinetosome, and the ribosomes are neatly packaged by a double membrane. Lots of aquatic and soil dwellers, many are parasites. We talked about Syncytium endobioticum, the potato blackwort culprit. What's fascinating there is how water availability can flip a switch. The same structure might release asexual zoospores if it's wet, or sexual gametes if conditions change. It adapts its reproduction to the environment.
Smart survival strategy.
Definitely. Then there's Chytriomyces hyalinus, a common decomposer that breaks down chitin. It's a great example of sexual reproduction via rhizoidal fusion. Two nearby thalases just connect their rhizoids, merge their contents, and form a tough resting spore together.
Simple but effective.
Exactly.
Yeah.
And maybe Noacauscala ramosa, a polycentric one found in water. It forms this branching filamentous thallus and has very distinct ribosomal aggregations in its zoospores.
Okay, on to the blastocladial.
Right, order Blastocladiales.
Yeah.
Known for thick-walled resistant sporangia and zoospores with that prominent nuclear cap and a side body complex. The star here is Alomyces microgenus. It's a classic model organism. Its big claim to fame is a true alternation of generations, which is rare in fungi. It switches between a haploid phase that makes gametes and a diploid phase that makes spores.
Like plants do.
Sort of, yeah. And its sexual reproduction is chemically sophisticated. The haploid form makes male gametophaga, usually orange and smaller, and female ones colorless and larger. The females release a pheromone, a chemical signal called syrenin. It acts like a powerful attractant, drawing the male gametes right to them. The males release one called parison to attract females. The result? Almost 100% fertilization efficiency. It's incredibly precise chemical communication.
Microscopic romance guided by smell. That's amazing.
It really is. Then, still in Blastocladiales, you have Helomyces, sorphore, the mosquito parasite. The massive discovery there was realizing it's heteroecious.
Heteroecious meaning?
It needs two completely different hosts to complete its life cycle. In this case, a mosquito larva and a tiny crustacean called a copepod. For years, people trying to use it for biocontrol couldn't figure out why it wouldn't always infect mosquitoes in the lab. Turns out, they were missing the copepod part of the cycle. It was a huge breakthrough.
Wow, that must have been a puzzle. Okay, one order left.
Last one, the order Monoblaepharidales, a smaller group, mostly aquatic decomposers. Their zoospores have characteristic clusters of ribosomes, lipids, mitochondria, and this structure called a rhomposome involves somehow with a flagellum. The standout here is Monoblaepharis polymorpha. Its sexual reproduction is unique among fungi. It produces a large, non-motile female gamete, basically an egg, and a small motile male gamete, an antherozoid, like a sperm. The male swims or crawls to the female structure, enters, and fertilizes the egg. This oogamous reproduction, big egg, small sperm, is much more typical of plants or animals, not usually fungi. It really shows the evolutionary experimentation within this group.
That's incredible. Egg and sperm fungi.
Pretty much.
Oh.
So as you can hopefully see, even though they're tiny, chytrids are just this hugely diverse, incredibly significant group. Those unique motile cells, their complex and varied life cycles, their roles as decomposers and sometimes pathogens, even their partnerships in animal guts. Understanding them is key to understanding fungal evolution, ecosystem health, and even things like animal nutrition.
Absolutely. It's mind-blowing. Chytrids, so small, so often overlooked, but they hold these incredibly complex biological stories. They influence everything from our food crops to how cattle digest grass. It really makes you wonder, doesn't it? What other hidden biological worlds are just waiting out there right under our feet or maybe inside an animal somewhere, holding secrets that could completely change how we see life? Well, thank you so much for joining us on this deep dive into the really fascinating world of chytrids. We hope you feel a bit more clued in now and maybe a lot more curious about all the unseen wonders out there in the natural world. Keep exploring.